Good afternoon. I am Dr. Jeannie Garbarino, the Director of RockEDU Science Outreach at Rockefeller University, and it is my pleasure to welcome you back for our third session of the 2021 Talking Science webinar series, COVID-19, Insights and Overview. We are grateful to the Andreas C. Dracopoulos Family Science and Society Initiative for generously supporting this annual outreach initiative that is designed to introduce high school students and their teachers to the renowned research being performed here at Rockefeller by our world-class faculty. We've had amazing participation in our first two sessions of this year's webinar series, and we are thankful to every one of you for joining us. Today, we are thrilled to welcome more than 1,300 registrants, engaging students and teachers from 33 states and 10 other countries around the globe. Welcome to those of you who are joining us for the first time. If you've missed our previous lectures, they are now available to view on the Talking Science website. In addition to these lectures, the website also offers some additional engagement opportunities that were designed for this year's topic. This includes links to recommended articles, as well as activities like data visualization, which maps out best practices for how to look at large data sets in ways that are meaningful and accurate. We also provide a tutorial on how to code your own map using a software package within R called Shiny. Finally, we have MAVE, Memes, Arts, Videos, and Expressions, which asks you to channel your creativity by creating and sharing posts about each topic discussed during this year's Talking Science lectures. Just remember to tag us at #RUTalkingScience, so that's the letter R and the letter U, Talking Science, when posting on your social media pages, and your work will be displayed on our website for everyone to see. Again, all of these fantastic activities can be found on the Talking Science website. If you would like to learn more about the other science outreach opportunities that we at RockEDU offer, from our Lab Backstage program that connects classrooms to a Rockefeller scientist for tours and demonstrations, to our mentored research program for high school students, I would like to encourage you to check out the RockEDU website. This year's Talking Science webinar series is designed to discuss scientific research devoted to the devastating COVID-19 pandemic. First, we heard from Dr. Marina Kasky, who discussed how antibodies can be used in treatment and prevention of disease. In our last lecture, infectious disease physician and Rockefeller visiting fellow, Dr. Chibozo Anamchukwu, talked to us about the inequities that exist within our healthcare system and how science can be used to combat these disparities. Today, we are delighted to welcome Dr. Paul Bieniasz, who has spent much of his career studying the human immunodeficiency virus, or HIV, which is the virus that causes AIDS. Dr. Bieniasz will discuss his work, which has transformed HIV virology and provided new tools for the discovery of novel approaches to treatment and prevention of HIV infection. He will also describe how the technology developed in his lab for cloning viruses has contributed to the discovery of treatments. Viruses have been around much longer than humans. They've caused human disease and devastating pandemics throughout recorded history. But for most of that history, we had no understanding of their basic nature. That all changed in the 20th century. Today, we can identify viruses by their genes and even see them under the electron microscope. Remarkably, through the work of Paul and others, scientists can now study ancient, extinct viruses through genetic sequences they have left in human DNA. Though we may fear them as a cause of disease, viruses are literally a part of who we are. Viruses can also be employed for human benefit. They can be engineered to execute many useful tasks, such as delivering genetic material to cells for lab research and to treat disease. Crucially, viruses can also be used to build vaccines. So before we begin, as we did last time, we're going to play a little trivia poll game here on Zoom that will see how much you know about viruses and vaccines. I'm going to share a few multiple choice questions with you all, and I'd like for you to select which of the 4 answers to each question you think is correct. I'll read the question aloud and give you all about 10 seconds to make a decision. I know it's not a lot of time, but we got to fit this all into a program. If you don't know, just take a guess. Don't worry, failure is a-okay. All answers are anonymous. Then I'll share the results, displaying how many people voted for each answer, and I'll tell you the correct answer. Okay, so let's go to our trivia poll. And let's see, the first question is a virus is a small collection of genetic code surrounded by what? As you are answering this question, I just want to say that viruses are amazing. I know that we're in a pretty poor place right now, but their biology is just incredible. All right, so most of you selected the correct answer, which is protein code. All right, let's see what we have for the next question. Oh, I love this one. Viruses can be found everywhere on Earth. They outnumber bacteria by which ratio? This one you might just have to randomly guess. I'm not sure how many people might have this sort of in their back pocket, but let's see how many of you are getting this right. All right, so the most selected answer was 20-1. Unfortunately, whoever selected that is wrong. The correct answer is 10-1. All right, so let's see. Here's a good one. Which of these diseases is not a viral disease? AIDS, strep throat, measles, or smallpox? All right, let's see. I think most of you are definitely getting this one right. Okay, and strep throat is the correct answer. So well done to everybody who got it right. Feel free to drop in the chat if you are confused about the other three, and maybe we can have a chat discussion about that. All right, so let's do one or two more. Oh, this is definitely one of my favorite topics right here. What are viruses that only infect bacteria? So a phage, fomite, adenovirus, or super spreaders? Very nice, everybody's getting the right answer for the most part. The answer is phage, although Dr. Bieniasz, maybe you can correct us on whether it's pronounced phage or phage. I'm never sure how to exactly pronounce that. So let's go one more because I think these are really fantastic questions. So the first inactivated flu vaccine was created using the fertilized eggs of which animal? Chicken, salmon, ostrich, or trout? All right, overwhelmingly correct, chicken. Well done, everybody. All right, so now it is my pleasure to introduce today's speaker, Dr. Paul Bieniasz. As a virologist, Dr. Bieniasz is the head of Rockefeller's Laboratory of Retrovirology and is an investigator at the Howard Hughes Medical Institute. Hailing from England, Dr. Bieniasz graduated from the University of Bath with a BSc in biochemistry and completed his Ph.D. at St. Mary's Hospital Medical School at the Imperial College London, University of London. After doing his postdoctoral research at Duke University, he then came to Rockefeller University. The AIDS virus, much like the novel coronavirus, has deeply affected humankind. Although antiviral drugs have significantly helped bring HIV under control in many nations, AIDS is still critically fatal in the developing world. AIDS caused more than 700,000 deaths globally in 2018 alone. Focusing on HIV, Dr. Bieniasz has spent much of his career trying to better understand how viruses replicate within their hosts and how they react when under attack by the immune system. With his partner, Dr. Theodora Hatzianu, he created an alternate form of HIV that has been used in animal models to mirror HIV infection in humans. The techniques Paul and Theodora developed to manipulate viral genomes proved essential when, in March 2020, they took the challenge of identifying ways to treat and prevent SARS-CoV-2 infection. Dr. Bieniasz's lab has made numerous contributions to our understanding of how the novel coronavirus infects human cells and how the human immune system responds to infection. For example, he has used engineered virus to create rapid, simple tests, assays to measure the potency of neutralizing antibodies isolated from the plasma of patients who have recovered from COVID-19. He is also a member of the team that developed the novel monoclonal antibodies for the treatment and prevention of SARS-CoV-2 infection. In mid-January, the university launched a first in-human phase 1 clinical trial of these very potent neutralizing monoclonal antibodies to evaluate their safety and effectiveness against SARS-CoV-2. Dr. Marina Kasky, who we heard from several weeks ago, is playing a key role in running this FDA-approved trial, which is being conducted in collaboration with the Bristol-Myers Squibb at the Rockefeller University Hospital. And on February 3rd, 3 Rockefeller University and Bristol-Myers Squibb announced an agreement that gives the pharma company exclusive rights to develop, manufacture, and market the neutralizing antibodies discovered at Rockefeller for the treatment and prevention of COVID-19. Meanwhile, Dr. Bieniasz is also contributing to our understanding of immunization as a key medical strategy in the ongoing pandemic. Members of his lab and Rockefeller professor Michelle Nussensweig's group recently completed a fascinating patient-based study of antibodies elicited by the Pfizer and Moderna vaccines, evaluating their effectiveness against variants of SARS-CoV-2 that were first detected in the UK, South Africa, and Brazil. Dr. Bieniasz's broad-ranging research has made him a go-to expert for journalists reporting on the evolution of SARS-CoV-2 and the biology underlying vaccine-induced immunity. His investigations will continue to provide information critical to our understanding of how COVID-19 vaccines and therapeutic antibodies work. As we face a mutating virus and the threat of resurgent infections, we need his work more than ever. We are really extremely excited to welcome Dr. Bieniasz as he presents Viruses and Vaccines.
Well, thank you very much, Jeannie, for that very kind introduction. Great pleasure to talk to you today, although this type of talk to what I anticipate is a very diverse audience presents something of a challenge, knowing what level of sophistication to pitch this material at can be quite difficult. So we're going to cover quite a lot of ground today, from sort of real basic fundamentals about viruses all the way through to some highlights of what we've recently published and even a little bit of unpublished research about SARS-CoV-2 and one other virus that we have been working on. So let's begin at the beginning, understanding viruses. And so what is a virus? And I was pleased to hear that most of you understand that what a virus consists of fundamentally is a piece of genetic material surrounded by a structure. So here is an electron micrograph of the virus that I've spent most of my career working on. That's human immunodeficiency virus. And on the right is the same virus represented in cartoon form. Obviously, it's a little clearer in cartoon form. So let's look in a bit more detail about what we have in HIV and generically in a virus particle. So all viruses have genetic material, and it can be in the form of DNA or its close relative RNA. Very similar molecule, just a little bit different, has more or less the same bases, but sufficiently different that it impacts how virus replicates. There are a variety of viral proteins that are included in a viral particle. One of the most important proteins we call the capsid. This is typically a quite structured arrangement of proteins that gives the particle its shape, its morphology, its intactness, as it were. Now, some viruses have a skin, a sort of a fatty layer around the outside that we call the envelope. So there are many envelope viruses. There are also many non-envelope viruses. The viruses we're going to be talking about today are envelope. They do have this sort of fatty layer around them. And embedded in that fatty layer are what we call envelope proteins. Those envelope proteins can be called things like spike in the case of SARS-CoV-2, but basically what they do are studs in the outside of the virus that allow it to attach to and enter into cells. Now, before we get into virology, a question that I'm often asked, and sometimes I think this is a fascinating question, and sometimes I just roll my eyes when I'm asked it. Are viruses alive? And the bottom line is there's no yes or no answer to it. Really, viruses have some of the characteristics of living things, but not all of the characteristics of living things that we encounter in our daily lives. So as we've heard, they have genetic material, they replicate, they make copies of themselves, and in so doing, they occasionally make mistakes, and we're going to go into that a little bit. And because of that, they can evolve through natural selection. One of the fundamental properties of living things they can do. Okay, but unlike most living things that you encounter, they're not made of cells that grow and divide, and they don't do metabolism. They don't turn food into energy or capture sunlight or any of those sort of more sophisticated things that living things do. So in a sense, you can sort of think of virus particles almost like the seeds of a plant, right? On their own, they don't really do anything, but once they get inside a cell and are allowed to replicate, then they take on many of the features of living things. So viruses come in many shapes and sizes. Here's a cartoon representation of a bunch of different viruses. So this little guy on the bottom right, parvovirus, that can cause some diseases in your pet dogs. This guy on the upper left, variola virus, that was the cause of hundreds of millions of deaths through smallpox. This snake-like creature here is Ebola virus that I'm sure you've all heard about. And the two that are perhaps uppermost in our minds in terms of public health significance at the moment, HIV-1 here, and then SARS, the original SARS virus that infected humans in the early part of this century, is a quite close relative of the virus that we're now particularly concerned about. So these viruses, some of them have DNA genomes, some of them have RNA genomes. They have all sorts of shapes and sizes. They're evolutionarily, many of them are unconnected to each other. They probably arose separately. And so while we think of viruses as sort of one category of biological entities, they're extraordinarily diverse. They're as different as you are from a jellyfish, for example. They're, in some respects, even more different. So a really remarkably diverse set of biological entities. So as I've mentioned, viruses by themselves don't really do very much. They really need a cell in order to replicate. And so what you can see on the slide in front of you is sort of a replication cycle for a particular virus, but I'd like you to think of it as sort of a generic replication cycle. And at its core, what happens is that a virus particle attaches to and enters into a cell, okay? In so doing, it delivers its genetic material into the interior of the cell. In this particular example, it's RNA, but for many viruses, it can be DNA. For some viruses, that RNA is then copied. In the particular case of HIV, that RNA is copied into DNA, which then enters the nucleus of the cell, the place where we keep our DNA, where cells keep their DNA. And then that DNA can then be copied again into RNA, leave the nucleus, and then that RNA is translated to make viral proteins. And the viral proteins are then all packaged together with the new viral genetic material and then leaves the cell as new viral particles. Now, some viruses sort of skip this DNA phase of the life cycle. They have RNA genomes. They totally ignore the nucleus, and their RNA is just copied to make more RNA. So SARS-CoV-2 is one example of a virus that does that. Other viruses like HIV go through a DNA intermediate through the nucleus. There are all sorts of replication strategies inside the cell, but the fundamental property of what happens during virus replication is typically one virus particle will enter one cell and perhaps hundreds or thousands of viral particles will be generated by that infected cell. So it's not just a replication system, it's also an amplification system. And so in a typical HIV infected person, one person can be infected by one virus particle, one cell is infected. 2 weeks later, there can be billions of viral particles in that individual. So a replication and amplification system. The other thing that can happen during replication is that the virus will make mistakes, okay? In copying the sequence of nucleotides that make up its genome, in the case of HIV, that is about 10,000 nucleotides in its genome. And on average, each time it copies the genome, HIV will make about one mistake. Okay, so a typical virus particle that's coming out of an infected cell, and I've marked them here by asterisks, on average, you'll get about one mistake per replication. And so these changes in the viral genome will obviously be translated into differences in the viral proteins. Now, those changes for the most part are inconsequential, or they might even be deleterious for virus replication. But in some instances, typically quite rarely, they're quite advantageous to the virus. Let's say this red mutation might allow this virus, for example, to more efficiently attach to a virus receptor, okay? In so doing, that mutation will increase the probability that that virus will then go on to make more copies of itself. And if that's true, then in subsequent replication cycles, that mutation can become more common in the virus population and over time dominate the population of viruses that, that is replicating in an individual. So viruses undergo Darwinian evolution in very much the same way that other that living organisms do so. But the pace at which virus replicates means that the rate at which viruses evolve can be very much faster than the evolution that you will have heard about that occurs over millions of years. In the case of viruses, and I'll show you some examples of this in a few minutes, we can observe Darwinian evolution in virus populations in a matter of days. Okay, so a very much faster cycle of replication than with living cellular life. Okay, just a brief general word about viruses and disease. So of course, the viruses that cause human disease are the viruses that are the most famous, but every minute of every day, all of us are infected by a number of viruses. They're replicating in our cells, evolving, adapting, being transmitted from one person to another. And many of those viruses, you just don't know they're there because they don't cause you any harm or any disease. So what's really the difference between a virus that causes disease and a virus that doesn't cause disease, and what accounts for the different types of disease that viruses cause? Well, that's a pretty complicated question, but it can be boiled down largely to a simple question of what kind of cells does a virus infect, okay? What happens to those cells once they're infected, okay? Do they live or do they die? And how many cells, what fraction of the cells in a particular tissue or body does the virus infect? And how well is the organism able to replace those cells if in fact they're killed by the virus? So a set of criteria. And so let's take a couple of examples. So HIV, for example, infects a particular type of cell in the immune system, the CD4 positive T cell. This is a cell that's crucial for orchestrating immune responses. And it kills those cells. It kills those cells quite effectively. And so over time, the number of CD4 cells in a person that's infected by HIV is diminished. And that really causes huge problems for the functioning of the immune system. And so the disease that's associated with HIV takes a long time to develop because we have reserves of these CD4 T cells. But once they're depleted, then our immune systems start not to function, and then opportunistic infections occur. And that's a very characteristic disease associated with that virus. A different virus, norovirus, right? Norovirus is a virus that you catch from food where people haven't washed their hands very carefully. That's a virus that infects the cells that line the gut, and it really replicates incredibly fast, tears through those cells, makes a big mess of them. And so you feel very sick very quickly, but your immune system is very well able to deal with that virus. And so you have a very large amount of virus very quickly, and then the virus is cleared away again. So you feel for a day or two like you're going to die, but then you recover quite quickly. And it really has only affected the lining of the gut. The rest of your body is more or less intact. And so the sort of biological footprint of viruses in terms of whether and how they cause disease is fundamentally affected by the types of cells they infect and how those cells survive and recover in response to viral infection. Okay, so let's move on a little bit to talk about something that's a bit more sophisticated, and an item that Jeannie mentioned in the introduction is how we engineer viruses to do things that are useful for us. So I'm going to take an example here. This is a virus called vesicular stomatitis virus, VSV. It's a virus that we like to use in the laboratory. It's sort of a model system virus. Really doesn't infect humans, or when it does, it causes either very mild or even not noticeable diseases. It's a virus that infects hoofed animals and also sandflies. It has a sort of a cycle between mammals and sandflies. These are biting insects that will, for example, bite a cow, become infected with the vesicular stomatitis that was in that cow, go off and bite another cow, and it spreads around in that way. It has an RNA genome, and it's very simple. It has 5 genes. Those genes can encode the outer membrane protein, the envelope or spike protein of the virus. That's encoded by this gene, the G gene here. And it's a virus that we like to use in the laboratory because it replicates very quickly, and we know how to engineer its genome to do useful things. So for example, I'm going to show you a movie in a second where we've simply added a gene to this virus. It's a gene from a jellyfish, a fluorescent jellyfish. And what happens is that when cells are infected with a virus, this virus where we've added this green fluorescent protein, then the infected cells go green, and you can watch the virus spread through a cell culture. Other investigators have taken this virus and changed the outer envelope protein of this virus from its normal, what we call G, the VSV-G protein, and they've replaced it with the equivalent protein from Ebola virus, a virus that I'm sure you've all heard about, caused devastating disease in West Africa a couple of times within the last decade. This virus here, exactly as you see it in front of you, that has been used to make a vaccine for Ebola virus, and it actually works very nicely indeed. At the bottom here is a virus that we've made in the laboratory where, again, we've changed the outer envelope protein of the virus. And this is now an envelope protein from an ancient virus, an extinct virus that we reconstituted by looking at old sequence and pieced them back together again to make a functional envelope protein. And then we combined that with an insertion of the green fluorescent protein into the viral genome. So we can both have the virus replicate in a manner that's dependent on the interaction of this envelope protein from HERV-T, but we can also see those cells being infected. So let's just watch a movie of VSV carrying GFP replicating through a monolayer of cells. So what you can see here on the left is a layer of cells that are sitting in a cultured dish. And on the right, what you can see is we've taken that same layer of cells and we've shone a laser light onto it that makes that green fluorescent protein glow. Let's play that again. And what you can see is at the beginning of the movie, there are very few cells infected. But as the movie plays, the virus is spreading from cell to cell to cell. And this entire movie takes place over 24 hours. So this is a very rapidly replicating virus, and one that we can watch its infection. We can quantify its infection really by measuring the amount of green fluorescent protein. So one of the things we're doing in the laboratory is to try and engineer a virus like VSV to kill cancer cells. And the way we're doing that is to take the HERV-T envelope protein, replace the VSV protein with that envelope protein. And it turns out that this HERV-T envelope protein likes to attach to cells that have a particular receptor on it. And this particular receptor is called MCT1. It's a protein that helps move metabolites in and out of cells. It turns out that cancer cells, certain types of cancer cells, have very high levels of this receptor. And because of that, this engineered virus is very good at infecting this particular type of cell. And it's very bad at infecting cells that don't have high levels of this receptor. And so what you can see on this set of images here are cells that we've infected with a virus built like this. And we've infected it with various dilutions of a virus stock. So here we've added about 3,000 times less virus, and we're still getting some infected cells. And when those cells get infected, they get ripped apart by this virus. Now, if we compare that to a cell that has normal, low levels of this MCT1, even the undiluted virus is very bad at infecting these cells. You can just see one little infected cell here. So what we are hoping to do is to use viruses like this to develop so-called oncolytic therapies, viruses that specifically infect and kill cancer cells. Okay, so that's one example of how we can use engineered viruses. Okay, so let's move on now. Let me just take a little drink of water to talk about immunity to viruses and vaccines. And so as I've told you, viruses sometimes cause disease, not always, but certainly sometimes. And because of that, and not just viruses, but all sorts of invaders that cause disease in hosts, we have a set of immune activities whose job it is to control the replication of pathogens, including viruses. And so one part of that immune system is comprised of B cells, okay? B cells are a collection of cells that make antibodies. We have millions of different kinds of B cells circulating through our blood and lymphoid tissue. And what happens is when we're infected by viruses, a subset of those B cells will recognize that virus. And they recognize that virus by virtue of having what we call B cell receptors on their surface. These B cell receptors are recognize individual viral proteins. And when they do so, that causes the B cell to grow and divide, keep growing and dividing for a little while, and then stop growing and dividing. And then these molecules on the surface form what we call antibodies. These, in very much the same way that the antibodies can recognize the viral proteins that they've encountered that's caused the cells to divide, those antibodies can also recognize the viral proteins, okay? And that essentially prevents the virus from infecting new cells, provided you have the right kind of antibodies. Antibodies work in another way. They can also recognize virus-infected cells, coat those infected cells. That really counts as a flag that tells other parts of the immune system to come and kill and eat those cells. Okay, so that's basically how antibodies are made and how they work. Now, as I'm sure you're all aware, in recent months, we have been, as a global population, afflicted by this new virus in the human population, SARS-CoV-2. And so we're going to spend the next few minutes talking a little bit about one of the proteins of SARS-CoV-2, the spike protein. So here is SARS-CoV-2, and it's studied on its outside by this spike protein. This is a close-up image of the spike protein solved by cryo-electron microscopy. The important parts of the spike protein that I'd like you to recognize are this blue part that we call the receptor binding domain. This is, or RBD, this is the part that attaches to receptors on the target cell surface. Those receptors are called ACE2. There's another important part called the NTD that's also quite important, and both the NTD and the RBD are really the important targets for neutralizing antibodies. The spike itself is composed of 3 subunits, 3 identical subunits, and only one of which is colored in this picture. So the gray, blue, and green parts form one of this trimeric spike complex, which is a complex of 3 identical subunits. So when SARS-CoV-2 started to infect people in the United States almost a year ago now, what we decided to do that we thought would be quite important was learn really how to measure the activity and potency of neutralizing antibodies that could interfere with the replication of SARS-CoV-2. We sort of recognized that antibodies were going to be a crucial part of how we get through this pandemic, be those antibodies elicited by vaccination or used as treatment, as we'll discuss. And so the way we did that was really to generate another engineered virus. This is a virus that doesn't have its own envelope or spike protein. We take the genome of that virus, HIV-1 in this case, remove its envelope protein, and then we make that virus in cells. At the same time, we put into those cells separately the SARS-CoV-2 spike protein. So what we can in fact do is make an HIV-1 particle, or population of HIV-1 particles that have the SARS-CoV-2 spike protein on their surface. So the outside of those virus particles looks like SARS-CoV-2, but inside they're HIV. And we put into that viral genome what we call a reporter, something like a green fluorescent protein, so we can easily count the number of infected cells. So we take that virus, put it on cells that have the virus receptor, and we put the mixture in the little wells of a microassay plate. And then we add antibodies to those microwells to see how well they're able to block infection. And that's really, in simple terms, how we measure neutralizing antibody activity. So here's some real data. These are 450 New Yorkers who donated their blood to have their neutralizing antibody titers measured. And they're arranged on the x-axis of this graph from left to right in order of potency. So the most potent neutralizers are on the right, least potent on the left. And on the y-axis is really how good their plasma, their blood plasma was, the source of antibodies for neutralizing the virus, so-called NT50 values. And what we found is that actually a rather small number of people are really very good at neutralizing the virus. They have lots of antibodies in their plasma, while there's quite a significant minority that have very low levels of antibodies. These are individuals whose plasma is very bad at neutralizing the virus. And these are the people that we think would be most at risk from reinfection. Fortunately, people who have had vaccines tend to be towards this end of the distribution. They tend to have better antibodies, more antibodies than those that are at this end of the distribution of natural infection. This is one of the instances where vaccination seems to be better at protecting people from infection than does prior infection. That's not always true for viral infections, but it does appear to be the case in this instance. So what we have also done in collaboration with investigators here at Rockefeller, and Marina Kasky told you about this work a few weeks ago, is to clone individual B cells from people who have been infected. And each individual B cell makes a different kind of antibody. So by choosing lots of different B cells that had made SARS-CoV-2 neutralizing antibodies and screening those antibodies, we could identify antibodies that were really very potent. So here on the y-axis, we're measuring the amount of infection, and on the x-axis as a function of the concentration of individual, what we call monoclonal antibodies. And working with Marina and Michelle and their team, we were able to discover very potent antibodies that are now in clinical trials. Now, one thing that we very quickly recognized that it was very important to understand how SARS-CoV-2 was going to evolve to become resistant to antibodies. Inevitably, as viruses evolve through populations, in many cases, they learn to adapt to the presence of antibodies. That process of mutation that I told you about earlier, that is basically how viruses manage to diversify, and some of those mutations will confer resistance to particular antibodies. So we developed this system, again using an engineered virus, to try to understand how antibody resistance might evolve. So this again involves an engineered virus. This time we have that same VSV virus that I told you about, but instead of the envelope proteins that I've told you about previously, what we did was put in the SARS-CoV-2 spike protein and green fluorescent protein. And then what we did was to grow that virus in cell culture for a few days. What that does is allow mutations to accumulate in the viral population. And then we take that population of viruses, and we estimate that we have about 1 million different variants in that population, mix those viruses with antibodies, monoclonal antibodies or plasma, and then add that mixture to cell culture. Okay, so virus mutants that have learned to resist, to avoid antibodies that are present as the cloned antibodies that we found or as plasma, they will then be able to infect cells, while those that have been neutralized by the antibodies will not. And so we're applying a selective pressure that enriches for these antibody-resistant viruses. And so here's some real data. So here's a cell culture dish that we've infected with 1 million different SARS-CoV-2 variants, that recombinant virus with the SARS-CoV-2 spike protein, in the absence of antibody, and then in the presence of one of the candidate and potent monoclonal antibodies developed for therapy. And you can see that it's mostly blocked infection, but you'll find little patches of cells, very much like the plaques that I showed you earlier, those little growing areas of infected cells where the antibody failed to neutralize that virus. And those represent viruses that have mutated to escape the effects of the antibodies. And here are some examples of those antibodies here. Okay, so here are some antibodies C121 and C144 are particular antibodies that were isolated from convalescent individuals, and C135 is a different antibody. And all three of those antibodies, if you look on this graph, are able to nicely neutralize the starting, or what we call wild type virus. Very small amounts of virus can reduce the level of infection to close to 0. But after we grow the virus, as I've just shown you, in the presence of these antibodies, what we get are mutant viruses. Here's one example, Q493R. Here's another example, R346S. This one, these two antibodies fail to inhibit that viral mutant, but the other antibody is perfectly good at neutralizing that virus. This mutant virus, the other antibody fails to neutralize it, but the other two are perfectly good at neutralizing that virus. And so what this shows you is that this particular recombinant artificial virus can learn to adapt to the presence of individual antibodies. And by learning where these mutations are, we can find out exactly how the virus can evolve to avoid those antibodies. Now, we've now done this with a rather large number of antibodies. Those antibodies are now listed on the left here of this rather complicated table, okay? And on the top is a list of viral variants, okay? And so here is K417. This is a particular position in the spike protein of SARS-CoV-2. Here's another one, E484, that's a different position in the viral spike protein, and here's N501. And what we have done in this experiment is to take the virus that was propagated in the presence of these antibodies and determine its genetic sequence. And what we find is that when we do this with particular antibodies, then we see that the percentages of viral sequences with these indicated mutations increases in the presence of particular antibodies. What this means is that the antibodies are putting a selective pressure on the virus that enriches for these mutations. And these particular mutations here, here, and here correspond to the viral mutations that have recently arisen in places like Brazil and South Africa and in the UK. These are clearly mutations that appear to be generated in response to antibody pressure. So everywhere where you see a red here, these are viral mutants that are arising in response to selective pressure imposed by viruses, okay? So we can look at how antibodies actually interact with the spike protein using structural biology. This is done by our collaborator, Pamela Bjorkman at Caltech. And what we find is that many of the antibodies that are generated either in people who have been infected or who have been vaccinated, if we select viral mutants that are resistant to those antibodies, and then solve structures, we can find that it's really the places where the antibody touches the viral spike protein where the mutations arise that cause resistance, okay? So this is something that's going to be an issue, something that's we're going to have to track in the foreseeable future. We've now done quite a lot of work on this, and everywhere where you see on this representation of the spike protein a little red spot is a place on the spike protein where we now know that antibodies bind and where they're capable of imparting selective pressure. These little blue blobs here focus on the receptor binding domain. These again are places in the receptor binding domain. The colors indicate places where antibodies impose selective pressure and where we can have mutations arise in our cell cultures that really tell us where the antibodies are putting selective pressure. Okay, so the bad news is that we can already see in nature these mutations starting to appear in naturally occurring variants. I've mentioned the Brazilian, the UK, and the South African SARS-CoV-2 variants that clearly show some of these mutations that indicate they're acquiring some degree of resistance to antibodies. The good news is that there are so many different kinds of antibodies that the impacts of these individual mutations is really quite modest. So here we're measuring the levels of antibodies that are capable of neutralizing the starting virus, and then a virus that has 3 of those antibody escape mutations. These are 3 mutations that are found in this particular example in the viral variant that's circulating in South Africa. And while you can see that for some individuals, there's a clear drop in potency for these neutralizing plasmas, these are mixtures of antibodies, most of those plasmas still have quite a lot of neutralizing activity against the mutant, the variant form of virus that is now circulating in those locales. And so, as I say, this is something that we're going to have to track and keep an eye on as vaccines are deployed and as the virus evolves and adapts to those vaccines. Okay, let me very quickly summarize as I've gone a little over time here. So we talked about the anatomy of a virus. It's really a simple packet of genetic information surrounded by a protein and sometimes a lipid skin. We've learned that viruses have some of the features of living things, that in particular that they can mutate and adapt. Some can cause disease, and I hope you've learned how we can engineer how viruses for various purposes, including making vaccines. Then we talked about how antibodies that are generated by B cells in response to infection or vaccines, they're a very important component of immunity to viruses, and as we're doing here, they can be used to make medicines. Inevitably, viruses such as SARS-CoV-2 can adapt to avoid individual antibodies. The most common antibodies they learn to avoid first. But of course, humans make many different types of antibodies in response to infection or vaccines. So as the virus adapts, there will still be antibodies that keep our vaccines effective, at least for a time, until we're able to make new types of vaccines to combat those changes. Okay, let me finish by acknowledging the many people that we've worked with over the last year, particularly on the SARS-CoV-2 project. So as Jeannie mentioned, I work very closely with my wife, Theodora Hadzianu. She and I lead the laboratory. I would like to particularly mention Frauke, Fabian, and Jiska, who really led the effort on helping to identify potent monoclonal antibodies from SARS-CoV-2 convalescent individuals at the outset of the lockdown last year. They were helped by Magda, Fengwen, Daniel, and Justin. But our efforts on this have really been just a component of a larger effort that's involved investigators at Rockefeller, particularly Michelle Nussensweg, Marina, who you heard from a few weeks ago. Other collaborators are Charlie Rice, again here at Rockefeller, Pamela Bjorkman and her team at Caltech, and then other investigators at University of North Carolina, New York Blood Center, and the National Health Service in the UK, who've really all together, this has been really a different way of doing science with a great deal of teamwork to try and understand immunity to SARS-CoV-2. Thanks
so much, Paul. We certainly do have quite a lot of time for questions, I think. And I think your last slide surely showcases how collaborative science is and the importance of collaboration in order to solve global problems. I mean, we're doing this right now at a pace, I think, that science has never seen before. And you know, I know that this is a very urgent type of case, but I do look forward to the shifts that we might experience in the scientific enterprise to really come together in these significant ways. Not that we haven't been always doing this, but it seems like it's been sort of, the paths have been cleared. So I want to start off with a philosophical question that was posed to us by Harry Fox. This is probably one of the harder questions that we can ask here, and that is if viruses are not truly alive, why do they exist? So I don't know if you want to discuss the existence of viruses in the first place.
I guess my retort to that would be, why not? So it is more or less true that viruses colonize more or less every living organism. You might find one or two exceptions if you try hard enough, but really what?
It's a hard one. Yeah.
Yes. I mean, basically they are a side effect of life, right? Life is replication, and viruses are just very simple replicating entities. As soon as you allow for natural selection, Darwinian evolution, all manner of biological self-replicating entities will arise. And that's not just viruses. There are viroids, pieces of RNA that parasitize viruses. They use virus replicative machinery to make copies of themselves. The minute you allow for replication and evolution through natural selection, all bets are off in terms of what kind of replicating systems can arise. And viruses are just many different examples of self-replicating systems with vastly varying degrees of simplicity and complexity.
Thanks. Joanne asks a more specific question around antiretrovirals. And she wonders if the antiretrovirals used in HIV would be effective against SARS-CoV-2, and why or why not?
Okay, the answer is no. And really the reason for that is that SARS-CoV-2 is quite a different virus. So if we just go down we'll take a couple of examples of antiretrovirals that work against HIV. So one major class is reverse transcriptase inhibitors. So reverse transcriptase is an enzyme that copies RNA into DNA. Okay, it's really a special feature of retroviruses. SARS-CoV-2 just doesn't have a reverse transcriptase. Its major replicative enzyme is an RNA-dependent RNA polymerase. So it copies RNA into RNA. And those two enzymes are sufficiently different that it's really, I would say, nearly impossible, perhaps not totally impossible, but very difficult to get a molecule that would inhibit both. And of course, when you're looking for drugs that are exquisitely potent and specific to HIV, then the chances of getting a molecule that inhibits SARS-CoV-2 are infinitesimally small. Same sort of true for classes of drugs like protease inhibitors. Both viruses do have proteases, but the proteases that HIV has are very different to the proteases that SARS-CoV-2 have. There was a suggestion early in the pandemic that one HIV protease inhibitor might inhibit a SARS-CoV-2 protease. It does, but it's so incredibly weak that it's not useful as a drug. So they're very different viruses with very different molecules, and it's just there's almost no relatedness between the two viruses. So it's just almost inconceivable that a molecule developed for HIV would work against SARS-CoV-2.
Thank you. We have another question around the elements of natural selection that you described for the viruses and the sort of emergence of these variants that are taking place. And so since viruses can evolve and mutate, does that mean we have to keep up the production of new vaccines similar in ways that we do to antibiotics?
It's very likely that will be the case. So viruses evolve at different rates. And let's just take two extreme examples. measles, right? We have a vaccine. We've never had to change that vaccine. It gives lifelong protection, near lifelong protection to everyone who gets it. Other end of the spectrum, we have HIV, right? That virus evolves so fast that if you take one virus from one individual and take the antibodies from that individual at the same time, the virus has already evolved to avoid those antibodies. Something sort of in the middle is flu, right? So that again, we have vaccines that work okay, not great, but they work okay, and we have to continually update them. We thought at the beginning that coronaviruses would be more towards the measles end, but it's beginning to turn out that they might be somewhere between measles and flu. Okay, so we're already beginning to see that some of the variants have a little bit of resistance to the vaccine-induced antibodies, suggesting that the vaccines are going to work pretty well for the foreseeable future. But a year or two from now, it's likely we're going to have to be modifying them, updating them to match viral evolution. That's a bit of a guess, but sort of the timescale that we're going to be thinking about, I think, in terms of vaccine updating.
In your talk, you discussed how the vaccine actually offers a higher level of immunity compared to being reinfected. Was that just a happy accident, or were scientists able to really zone in on the mechanism behind that and as a means to create a strong vaccine? That's really an accident of the virus biology. So what we find with SARS-CoV-2 is there's such a very large range in the magnitude of the immune response that it was almost inevitable that vaccines would only comprise a subset of that. What we think happens is that when people become infected with SARS-CoV-2, there's such a wide variety in terms of the amount of virus that replicates in an individual. So some people are completely asymptomatic. The virus replicates a bit in their nose and they never even notice it, okay? Those people tend to not have mounted very large immune responses. Other people might get a heavy cold, have a lot of virus in nasal cavities in the throat. They probably had a bit more virus. And then very severe infection when the virus gets down into the lungs and it can disseminate systemically, then the immune system has experienced rather a large amount of virus. And so those people tend to have stronger immune responses. So there's a massive variation in SARS-CoV-2. Most people have very mild disease. Now, when you have vaccines, you get pretty much a standard amount of virus antigen injected into a muscle. So scientists take steps to maximize the amount of immune response to that. So in vaccinees, the immune response tends to be more consistent. And so these people who have had very mild disease and very little immune response, they're just not represented in the vaccinated population, which is obviously a good thing. So it's really for that reason that vaccines give more consistent, more robust immunity than does natural infection. And adding a little bit onto this, you discussed the role of B cells, but we have a question from Mary that wonders, what role do T cells play? We're hearing a lot of that in the news. Yes, so
T cells very likely play an important role in immunity, but they have been sort of touted by some, how should we put it, less charitable members of our society who want to, perhaps deny the role of antibodies in protection and so on and so forth. Really, the main reason we focus on antibodies is because they're easy to study and we know that they can provide protection. We know from experiments that we can do in animals that if you take the antibodies from one animal, purify them, put them into another animal, they can protect, okay? those types of experiments are very much more difficult to do with T cells, okay? And so the level of certainty that we have that T cells are an important factor in immune protection, that level of confidence is just not quite there that we have for antibodies. In reality, when somebody mounts an immune response, the amount of antibodies they make and the amount of T cells they make, not perfectly, but tend to be broadly correlated. And so if you're looking for signs that somebody is protected and you measure antibodies, right, you're measuring something that you know is important for protection, and you're also measuring something that you have a pretty good idea will correlate with the amount of the T cell response. And so when we talk about correlates of immunity, antibodies are measured because they're, as I say, they're straightforward to measure and we know that they're important. We strongly suspect that T cells are important. They're just tougher to measure and tougher to show they're important.
Yeah, I assume that just more time is needed. It's
Yes.
amazing because science has been taking place at a pace that I've never, ever, I mean, I've only been in the field for 20 years. So, I mean, that's a bit. Am I saying that out loud? You know, I've never seen it move this fast. And I guess it's just that you know, given the studies and the technology that has been developed already really set the course for how effective we could respond to multiple elements of the pandemic. So I think that's incredible, which includes the possibility of Moderna developing a third dose. So we have this question from Toby that asks if Moderna is going to, we've been reading about the possibility of Moderna developing a third dose. Would this be different from the first two doses if it's intended to protect against these new variants that are popping up?
Right. So this is an interesting and important challenge. So while the news is full of stories about variants, it's quite important to realize that at the current time, in a global sense, these variants are actually still quite rare, okay? So that they're common in particular places across the world. But in New York, for example, we still mostly have the original or strains that are close to the original. So if I'm getting vaccinated tomorrow, I'm not, but I wish I was, but if I was, I'd want the original vaccine, okay? Now, when and how does, for example, Moderna make the decision to change the vaccine to a different strain? That's a tricky question, and we're going to have to watch and wait a little bit to see how and which variants are successful in spreading throughout the world. It might very well be that 6 months from now, we've vaccinated large numbers of people and these variants stay at very low prevalence and very isolated in particular areas of the world. If that's true, then there will not be a very strong impetus to change the vaccine. On the other hand, it might be that the South African variant outcompetes all others and dominates the global landscape. Then it'll be very clear what next year's or the next batch of vaccines need to be. But that's a little different to the UK strain. It's a little different to the Brazilian strain. So, you know, there's a little bit of preparing, okay? There's certain things that Moderna, Pfizer can do to prepare to make the next generation of vaccines. But really, to roll them out, you do have to wait a little bit and see what grows to sort of become next year's strain or 2 years from now strain.
Yeah, we've been talking a lot about the symptoms that we're seeing with viral infection, particularly with the SARS-CoV-2, with the cold and the headaches. But I think there are other types of presentations that a virus can elicit. And so we have a question from Jerome about the link between viruses and the immune response, our immune response to viruses and the development of cancer. I was wondering if you might want to just have a couple of comments around that link.
Right. So viruses are associated with cancers, and they can be associated in really fundamentally different ways. So HIV, for example, there are some cancers that are associated with HIV infection. What we suspect there is that it's really a failure of immune surveillance to tackle cancers in their early stages that you would ordinarily never know that you had or had the precursors to. And that, so an insult to the immune system can, in principle, predispose to cancer. There are other viruses, papillomaviruses, for example, that clearly are direct causes of cancer, because we know that because the virus genome is in the cancer itself, and we know that there are viral proteins that cause cells to grow and cause precancerous lesions. Okay, so there the result is very direct. There are other instances, and thinking more along the lines of things like hepatitis C, where there's clearly a causal relationship between the virus and the cancer, but it's less direct. And in that case, we don't really know, but what we suspect is that there's a lot of liver damage that's caused by the viral infection. And it's the process of regenerating liver, the liver cells having to go through many more cell divisions than they ordinarily would, that just increases the chance that they would acquire a mutation that might lead to cancer. There are all sorts of other mechanisms to do with inflammation that are actually quite complicated, and I'm not really sure that anyone fully understands the connections between viruses and cancer there. But yes, the connections between virus and cancers can range from the very direct to the, just having more cell division in response to viral infection being associated with cancer. And those are just examples. There are many more one could go into.
So how much did scientists have to examine the natural mechanism of entry for the SARS-CoV-2 virus as they designed the vaccine? For example, does the vaccine have a lipid bilayer? And if so, is that important for delivery of the vaccine?
So we had to know, I say we, I'm saying the royal we here being scientists in general, we had to know a little bit about the viral entry mechanism. But there was already a lot of knowledge in place based on other viruses. So fundamental studies with influenza, with HIV, with respiratory syncytial virus, other coronaviruses. This sort of trimeric spike structure is something that recurs through viruses. And there's a fairly well conserved mechanism by which those spike proteins on the outside of viruses cause the lipid bilayers around the virus to fuse with a target cell. So while there are peculiarities to coronaviruses, and they had actually been quite well studied by people like Jason McClellan and others, that allowed them to design mutations to stabilize the spike protein in a form that it is before viral entry and fusion, where you really want the neutralizing antibodies to target. Those studies were pre-existing, but in the case of coronaviruses, were kind of quite recently pre-existing. So they'd been working on vaccines for things like the first SARS and subsequently MERS, and could instantly translate that knowledge to design mutations in the SARS-CoV-2. There have been more recent developments that can make those things even better, actually, more stable, pre-fusion, as they're called, conformationally stabilized spikes. So it's a mixed bag. There was already quite a lot of knowledge that enabled that vaccine design to proceed at a pace.
Thanks. And I think looking at the time, I think we'll end with Mina's question, which is, why does COVID-19 produce such a wide range of symptoms compared to other viruses that infect humans?
That's a very good question, and I'm not sure I'm going to be able to give it a very good answer. Now, we know that the first types of cell that get infected are those ACE2 cells that have the ACE2 receptor in the nose, okay? And that in severe cases of disease, the infection proceeds to ACE2 positive cells in the lung. So it's pretty clear that most of the respiratory symptoms are due to infection of those particular cells and the immune response to those cells in those tissues. There's sort of respiratory distress that is characteristic of most, I hate to say, I would perhaps not say most, many cases of severe COVID-19 disease really can be traced to the types of cells that are infected in the respiratory system. Now, when you get into things that are more complex, like the clotting disorders and long COVID, there the pathogenic mechanisms are just not particularly well understood at the moment. They are quite likely to involve not just the cells that the virus infects. The sort of the simple model that I presented in the talk about the disease footprint being highly dependent on the types and numbers of cells that are infected. That's almost always true. In this case, there might be some extra complications, very likely some extra complications that it might take some time to figure those out. And I'm sorry that I don't have the answer for you now, but that's just where science is at this point.
Thank you. I think it's important that we also, for everything we know, there are still so many things that we don't know, and that's what keeps us employed, I suppose. Yeah. I do have one last question. It was asked what kind of plant is in your background. I thought it would be, as a plant lover, I would love to also know because it's lovely.
So this one here is a money tree. There's also a couple of them there. These things are very easy to cultivate, so I take cuttings every so often. There's some more money trees there. That one right at the back, I actually don't know what that's called. That was in our house, and my wife grew tired of it, so I brought it to work.
Well, they look great. I think you would get 10 out of 10 on that Room Rater app or whatever it is that they're doing on Twitter these days. Correct. Well, thank you so much for this wonderful talk. I think every time I hear about your work and the work of our colleagues who are also in virology, I often regret that I did not go into virology because it's absolutely stunning and incredible. So thank you.
Thank you for having me.
Oh, it's our pleasure. So everybody, thank you again for coming. I would like to remind you to continue checking our website for more information about our next session, articles of interest on each topic, interactive activities I told you about earlier, as well as videos of past Talking Science events. For this week's MAVE theme, we would like you to think about viruses and vaccines. Vaccines are a vital tool in preventing the spread of highly contagious viruses. What is the relationship between virus and a vaccine? Are there things in your life that spread quickly between people like viruses? What kind of vaccines are there to prevent those viruses? While we would love to hear your responses to these questions, please don't limit yourself to them. Submit anything related to Dr. Bieniasz's lecture that moves you. We look forward to seeing your creativity. Finally, we look forward to welcoming you all back for our final Talking Science session on Monday, February 22nd, 2021 at 4 p.m. It will feature Rockefeller University President Rick Lifton. This is going to be a truly exciting and unique opportunity for all of us, including me. Rather than a lecture, I will engage President Lifton in conversation to discuss the COVID-19 road to recovery. You will hear us talk about a wide array of topics, including public health, testing, vaccines, and how we can prepare to prevent future pandemics. You will also have the opportunity to ask President Lifton questions of your own during our Q&A session. We certainly hope you will all join us. Thank you once again for attending today's event, and have a lovely evening.